Okay, so just when you thought it was safe, I decided to make another video. So this is going to be an overview and include some of the Gizmo information on it. So here we go. Let's look at respiration. We know from the start, we've talked about catabolic and anabolic processes. So we know that when we're talking about metabolism, we say that we're bringing in some nutrients. Those nutrients come into the cell and they get metabolized. In other words, they get broken down through the process of some type of catabolic activity, and we release energy from that. And then we also will release some other things, some waste products, etc. And then the goal, of course, is to make that energy, and that's the big goal. And then the cell also needs to use energy, and it's doing that all the time simultaneously when it's making things, when it's doing active transport, all sorts of other activities driving endergonic reactions forward. So we got a lot of use of ATP, and we have a lot of need for ATP. So let's see where this happens. Now, one thing that we haven't talked about yet, but is in OpenStax too, is that one of the mechanisms for getting glucose into cells is using something called an insulin receptor. So here you see the insulin receptor, and here's the insulin. So in response to the presence of sugar or glucose in the bloodstream, insulin is released. Insulin then will go and on the cells find this insulin receptor, and it's going to activate this insulin receptor. And then one of the things that happens when it does that is that it's going to put a glucose transporter into the membrane. And so this GLUT4 is representing this transporter that now is going to, which was in this little vesicle here, and now is being moved to the membrane. So it's going to facilitate this glucose coming in. So that's kind of fun and valuable. Once glucose comes in, though, the cell wants to keep it there. It wants to capture it. And this is the first series of those 5 steps that we've seen in the cytoplasm that we know of as glycolysis. So glucose enters the cytoplasm, or cytosol. The first thing the cell does is capture it. And the way it does it is it uses the enzyme, hexokinase, together with ATP. So ATP is used here. ATP, the phosphate from ATP is going to be used. This molecule is going to release energy when it's hydrolyzed, right? Water comes in, splits off that phosphate, and gives off energy. And that energy is used to take the phosphate from ATP and put it here. Meanwhile, what we end up with is ADP. So again, glucose comes in. The enzyme hexokinase is catalyzing this reaction. This would be an endergonic reaction, so this wouldn't happen spontaneously, except it will, so long as we couple this reaction to this breakdown of ATP. And this reaction has a negative delta G. This reaction has a positive delta G, but put together, coupling, will get us to glucose 6-phosphate. Now the cell has accomplished something. Glucose 6-phosphate won't go back out. So it's kind of captured glucose this way in the cell. Now the second step of the reaction, we have a new enzyme. This enzyme is called an isomerase. So if you remember what isomers are, they're basically just a rearrangement of a molecule into a different form. So glucose 6-phosphate, in other words, it has the phosphate on the 6th carbon, is rearranged, still has phosphate on the 6th carbon, but now it's in the form of fructose. So that isomerase enzyme helps to rearrange this from glucose to fructose 6-phosphate. Okay, step 3. Guess what? We need to invest energy again to make this reaction happen. So here what you see is that fructose 6-phosphate is going to have another phosphate added onto it. And so by adding this another phosphate, we need the phosphate from ATP. This reaction, again, needs to release energy in order to push this reaction forward. the enzyme that's used is another one that we call a kinase. So you see this is a kinase, and this is a kinase. This is hexokinase, and this is phosphofructokinase. Now hexokinase, I actually want you to know that enzyme name. This one, well, it is important, but I'm not going to make you memorize all of these enzymes. So you can know, so you have my permission because it's a great phosphofructokinase. Just say it a few times and it becomes magical. So we have ATP being used, hydrolyzed, releasing energy, driving this reaction, which wouldn't go forward otherwise, forward. And now we have fructose 1, so there's the phosphate on the first carbon, 6, there it is on the 6th carbon, bisphosphate. And the bi or bis, it just means there's 2 now, 2 phosphates. So in this this is a little bit of a dancing step here. This molecule now is going to get split. And it's going to get split into 2 3-carbon compounds. This was 6 carbons. This one now has 3 carbons. This one now has 3 carbons. And the ultimate goal is that we're going to make not this one up here, which is this dihydroxyacetone phosphate. Don't worry about the name. But once this is made, it too is going to get converted into this. So we're going to get 2 of these out of this process. 2 of these called glyceraldehyde 3-phosphate, sometimes abbreviated G3P, and it has some other names too, but let's keep it at that. So this is our G3P. All right, perfect. So now we take that G3P, and this is the payoff phase. So going from 6 to 10, we're actually now going to extract energy. We're going to remove electrons. We're going to make some ATP to kind of replace some of the ATP we just used, but also a little bit more. And again, all of these are going to be enzymatically controlled. So we're going to have enzymes in all of this. So glyceraldehyde 3-phosphate, G3P, is going to have, it's going to actually be the agent that is going to add electrons and that hydrogen onto NAD+. So NAD+ in this form, remember, was oxidized. Now it's reduced. And where those electrons and hydrogen came from was this. So then we go to the next one, and we have this molecule here. And we're going to take this molecule, and notice what's happening. Look at the arrows. So in here, we're not using ATP, we're making ATP. This is called substrate level phosphorylation. So what we're going to use again is a kinase, because kinases have everything to do with putting phosphates on and off of things. And in this case, this particular phosphate, this kinase is going to take the ATP that's out in the cytoplasm, and it's going to add another phosphate on it, making ATP. Cool, now we got another step, so let's go to here. We have another step, and we go to here. We have an enzyme called a mutase, and we keep going. We remove some water, and here's another step where we make ATP. So for this one single G3P, remember we have 2, we're going to make an NADH, an ATP, an ATP. Do it again. So in the end, we're going to have 2 NADHs and 4 ATPs that are made in glycolysis. All right, next, link reaction. So the other thing, whoops, I didn't really talk about this other final product. So here's pyruvate. This is the end product of glycolysis. We have pyruvate. And pyruvate now is going to go out of the cytoplasm, and it's going to go into the mitochondria. So here it is. Pyruvate is a 1,2,3 carbon compound. It's going to get transported in, so it's a transport protein. And then pyruvate's going to get converted and as it gets converted, we're going to do a couple things. It's going to get converted to this, acetyl-CoA. We're going to add this coenzyme in. We're going to remove a carbon dioxide. And we're going to make another NADH for each pyruvate that goes in. Remember now, we have two, because we made two from one molecule of glucose. So this is called the link reaction. Some people just kind of incorporate it into the Krebs cycle, but I kind of like looking at it separately. Remember, this is the mitochondria. I thought it might be useful to see a mitochondrion, remembering that this is actually an actual electron micrograph. So it's a picture using a microscope of a mitochondria in a cell. And this is an artist version. So you can see here it has this outer membrane, has all this material in here is all this inner membrane. There are some spaces. Here's a matrix. There's a little gap between this inner membrane and the outer membrane. So these cristae are actually the folds back and forth, back and forth, back and forth. And it also has its own DNA. So we'll talk about this more, but this is just to give you some visual of what's now. We have pyruvate going in here into the mitochondria. Now what? So pyruvate was made into that molecule that we said was acetyl-CoA. So here's acetyl-CoA right up here. And acetyl-CoA is now going to enter this thing called the Krebs cycle. It has only two, because it lost one for carbon dioxide. It has two carbons, so there's 1, 2, this one, and then it has this CoA, but that's kind of like an attachment. Acetyl-CoA, 2 carbons, is going to join with this 4-carbon compound called oxaloacetate. I want you to know that one. Acetyl-CoA, oxaloacetate. We're going to, with of course enzymes everywhere here, but we're not going to talk about them, is going to form this molecule called citrate. Citrate has 6 carbons. Where did they come from? 4 from oxaloacetate, 2 from acetyl-CoA, form a compound called citrate, has 6 carbons. Now follow the carbons going from citrate, isocitrate, and we go, so this is 6, we still have 6. Where do we lose the carbon dioxide? Right here. Now we have only 5 carbons. Okay, so here we are counting carbons. So we go from 6 at the top of this cycle. Notice this one just says isocitrate, still has 6 carbons, so that must mean it's an isomer, right? Or just a rearrangement. We go down to this one, which is alpha-ketoglutarate. Again, you don't need to memorize that. But notice it's only 5 carbons because we lost a carbon in the form of carbon dioxide. But also, more importantly, look what happened right here. So at this stage, anytime you see these carbon dioxides going off, we also have a redox reaction happening. So something is getting, this isocitrate is getting oxidized, and then as a consequence, this NAD plus becomes reduced, meaning it's picking up the electrons and the hydrogen. Keep going. We have another step just like the one we just described above. Lose another carbon. Gain, that's interesting. Okay. So now we're at 4 carbons, sorry, not counting well, end of the day. And right here, we make an ATP. Now you say, uh-oh, that's not ATP, that's GTP, but this gets converted to ATP. So we actually create an ATP here. Keep going. We have another molecule here. Keep going. Here's the first place we make FADH2. It's actually the only place in this cycle, but we'll make it twice because we'll go around twice. We go to here, we go to here, we make another NADH. And now we're back to 4 carbons, oxaloacetate. It's going to pick up the next acetyl-CoA. So that's a pretty good stopping point before we move any further. So hang in there.